Good morning, everybody. Welcome to Discovery Day Online. I am Gabriel Santos, Collections Manager and Outreach Coordinator of the Alf Museum, and I am very, very excited to welcome all of you today to our Discovery Day Online event, The Mighty Dinosaurs, dedicated to all things dinosaurs. This morning, I'm very, very excited to welcome our special featured speaker, Dr. Catherine Early. Good morning, Dr. Early. How are you doing today? So today, oh, sorry, one second. I forgot to unmute you. Okay, so uh, it's a Saturday morning. I'm still trying to like get started.
I get it.
So, how are things going for you right now?
Going great. I'm excited to see spring come in, see some new birds coming through, and I'm really happy to be here.
Awesome. Well, speaking of birds, that's kind of what you're going to be talking with us about today, right?
Mm-hmm.
Awesome. Well, let me introduce all of you to our guest speaker. Dr. Catherine Early is the Barbara Brown Chair of Ornithology at the Science Museum of Minnesota, where she curates the biology department's 170,000 specimens. Her main research interest is the relationship between cranial anatomy, sensory specializations, and behavior in vertebrate animals, with a current focus on modern day and extinct birds. She earned her PhD in Ecology and Evolutionary Biology from Ohio University, where she used CT scanning to study the shape of the brains of birds and how they change through evolutionary time. So I'm really, really excited to talk a little bit about dinosaur brains. Are you gonna? Because like, a lot of people have that old perception, right? That dinosaurs have like those pea size or peanut sized brains and everything. You're gonna
Mm-hmm.
help us kind of dispel that myth a bit today, right?
Oh yeah.
Awesome. All right. Well, what's going to happen today, everybody, is the same as our usual Discovery Day Online thing. Dr. Catherine Early is going to give her presentation, and following, we'll have a discussion and Q&A. So if you have any questions, please put them in the chat, and we'll make sure we get to them. All right. You are good to go.
Okay. Well, thank you for that introduction, and thanks everyone who is joining us. I'm coming to you live from St. Paul, Minnesota, which is the home of the Science Museum of Minnesota, and I'm excited to be talking to you about my research. But before I get to that, I wanted to do a quick introduction of myself, get a little deeper than Gabe was able to go, and start from the beginning. So, growing up in North Carolina, where I'm from, I was a big nature nerd. I loved animals, and I got to indulge this at the North Carolina Museum of Natural Sciences, where I volunteered with them with their living collections. You can see one of their outreach snakes with me right there, and then continued to work with them in a lot of different capacities. Like I helped with their birthday parties, I worked in the gift shop, and I ended up coordinating a teen science cafe program as well. After that, my interests and a really excellent undergraduate mentor at North Carolina State University, where I did my bachelor's, led me to pursue a PhD in ecology and evolutionary biology at Ohio University. After that, I joined the Florida Museum of Natural History and the University of Florida as a postdoctoral researcher on the Open Vertebrate, or OVERT, project, which is an NSF funded, so National Science Foundation funded collaboration that is digitizing vertebrate specimens through CT scanning, which we are going to talk about a little bit later in my presentation. Now, like Gabriel Phillips said, I am the Barbara Brown Chair of Ornithology at the Science Museum of Minnesota, where I head the biology department and continue my research in ornithology. And my research answers the questions: how do the brains of birds reflect behavior, and how do they change through evolutionary time? So you should see on this slide popping up, hopefully the animations are coming through, that we've got the skull of a bird, and I've kind of blown up what we call a brain endocast, which I'll explain in a minute, but the representation of the brain. But you might be saying, wait a minute, this event is called Mighty Dinosaurs. I thought I would be learning about dinosaurs like Acrocanthosaurus up here. Well, in case you didn't know, scientists have found multiple lines of evidence, including skeletal anatomy, as illustrated here by Scott Hartman, to support the evolutionary hypothesis that birds are dinosaurs. So they are representatives of a certain group of dinosaurs called theropods. It also includes things like Tyrannosaurus rex. So I'm going to be focusing on what we call avian dinosaurs, or birds. And like Gabe said, we're going to dispel some of the myths about bird brains. So if you're someone who doesn't study birds or doesn't think a lot about birds, something like a house sparrow, shown here, might be your stereotype of what a bird is. Kind of small, capable of flight, and maybe using its beak to pick up things off the ground. But birds are a fascinating group of animals that could never be constrained by those simple definitions and descriptions. So they're the most speciose group of land animals, means they have a lot of diversity, with over 10,000 species known to be alive today. And this diversity means that some pretty interesting traits are represented. So for example, you've got these flamingos up here that engage in something we call filter feeding, and you can see in this video up in the corner, they're kind of digging around in the mud and using their really sensitive bills to filter out the non-food things like mud and water and keep in the food particles that they like to eat. There's also, and this is a pretty well-known trait for birds, a huge range of coloration, represented here by this picture of a lilac-breasted roller. And there's also some birds with some decidedly unbird-like traits, such as the nocturnal kiwi, who never has any hope of flight, and has a really sensitive bill tip, a really good sense of smell, and as you can see in this GIF, it is a little bit clumsy sometimes. They're one of my favorite birds. So some of this diversity in birds is reflected in the different behavioral traits of this group. So birds can engage in a lot of complicated display behaviors, or maybe a more common example you might think of male peacocks with that giant fan that they'll lift up to impress females. They engage in a lot of different locomotion styles. So you can have gliding birds like vultures, hovering birds like hummingbirds whose wings beat so fast that you can barely see them with the human eye, birds that kind of fly underwater like penguins, and birds that don't even bother with flight like kiwi I mentioned, and also like ostrich, who are still really good at getting around because they've got these really fast legs. Birds actually have a lot of intelligence. So I know bird brain used to be used as an insult, but especially certain groups of birds like parrots here are highly intelligent. So parrots in particular have a really well developed sense of language and can learn a lot of human letters and numbers and language. And then you also have a huge diversity in feeding. So one famous example of this is Darwin's finches that have beak shapes that are kind of adapted for the different types of diets they were using on different islands. Or this great illustration by Tyler Lang showing all the different beak shapes that you can, or a lot of the different beak shapes that you can see in the bird world. But ultimately, all of these different behaviors are governed by the brain, which is why I and so many other researchers are so interested in its evolution. And this is what really hooked me into research, because the kinds of questions I'm drawn to are the questions about the relationship between structure, so the shape of something, and function, so what purpose it serves or what it does. Now, we know from looking at the brains of modern day animals, this idea that's developed by Dr. Harry Jarrison, that the relative size of a brain structure correlates with the importance of the function it serves. So that means that a brain structure like this thing right here, which I will describe later, but it's called the optic tectum, which we know serves a vision purpose, is probably going to be relatively larger in birds that are more reliant on vision, or a lot more reduced in birds that don't need a lot of visual information to navigate their environments. And because of this relationship, we should be able to make inferences about the behaviors of extinct birds based on the shape of their brains, or maybe the size of different structures. Oh, and I should clarify, when I say the relative size of a structure, I mean the relative size, so the size of the optic tectum relative to entire brain. So maybe I need to clarify, you can have a huge range in size in birds. You can have tiny little hummingbirds and things like ostrich, who are like as tall as me, or even taller. So when we talk about the structure and function relationship, what matters is the relative size of that brain structure, not the overall size, because obviously any brain structure on an ostrich is going to be bigger than the same brain structure on a hummingbird. And just like all the other traits of birds that I was talking about before, their brains are just as diverse as they are because, like I said, they reflect that diversity of behaviors. But the diversity of modern birds, like this quail here, which you may recognize, didn't just arise out of nowhere. They're part of a lineage of non-avian theropod dinosaurs. So that was the group that I highlighted in that earlier slide that birds belong to. And there are millions of years of evolution between the oldest bird and the birds we can observe today. And so without understanding brains and behavior through deep time, we're missing a huge chunk of the evolutionary history of this or any other group. So we're missing the context of how birds evolved into what they are today. Now, you can't observe behavior in the fossil record. That's the way it is. You know, we can't look at these extinct non-avian dinosaurs and see what they were doing. But since we know that there's a correlation between the brain shape and brain anatomy and behavior, we should be able to study the brain. Unfortunately, also, in the fossil record, brains don't really preserve very well in 3 dimensions either. So we're left with this problem. How do you study brain evolution without a brain? So one way you can do it, and the way I do it, is with brain endocasts. So that's what I talked about on that first slide where I mentioned my research. A brain endocast, and for context, we've got this grouse. His skull is here, and then we zoomed it way in so you could see the brain endocast here, which represents the space in which the brain sat during life. So you can see this empty space over here. And in birds, brain endocasts are a pretty faithful representation of the space in which they sat. So in other groups of vertebrate animals, like fishes, for example, there's quite a bit of space between the brain and the brain case, so where the brain sits. But in things like birds, you have the brain, and then you have the bones of the brain case right up against the brain. And so you can see a really accurate representation of the shape of the brain. And so how do we get these brain endocasts? Well, one option, now that we live in an era of a lot of advanced medical technology, is to use CT scanning, which is like a 3D X-ray. If some of you may have actually had them for health issues before. You put your specimen, so I use museum specimens in my research, put that in the scanner. That generates a bunch of what we call cross-sectional images of the hard tissues of the specimen. So all this white stuff is the bone, and these spaces right here are where the brain sits. And then combine those. Oh, and yes, I should say CT means computed tomography, but it's easier to just think of CT scanning equals 3-dimensional X-ray, kind of. And then combine all these cross-sectional images to make a 3D model of the brain, which again is called a brain endocast. So we can study the behaviors of extant or modern-day species of birds, and we can study their brains with their endocasts. In fossils, we can only study the endocasts. So we need to figure out a way to make a connection between brains and endocasts so that we can infer behavior in extinct birds. And this is a pretty common practice to do in the field of avian paleontology, especially my subspecialization is called avian paleoneurology. One example of a study that did this was led by Dr. Maria Eugenia Leone Gold. She studied the brain endocast of a dodo. So I'm sure you've heard of these. If you ever watch the Ice Age movies, they figure prominently. This is also where the phrase dumb as a dodo came from. So dodos were a flightless relative of pigeons that evolved on an island where they didn't have a lot of predation pressure. So they didn't have a lot of things trying to eat them. And then when white people arrived, suddenly they decided to eat a lot of dodos. And the dodos were not adapted for avoiding predation, even including by humans. So they went extinct pretty quickly. And the people who caused their extinction thought, oh, well, they were just dumb. That's why they went extinct. But actually, Dr. Gold and her co-authors found that the brain endocast of a dodo really wasn't that different from the brain endocasts of their modern day relatives. You know, there were some differences because you can just look at the slide and see minor variations, but they weren't significant differences. And so a dodo is not really any dumber than a pigeon. And, you know, you might not hold pigeons in very high regard, but you have to keep in mind that pigeons have been able to thrive alongside the most dangerous species on the planet, that's us. So they clearly have what it takes to adjust to completely new environments. So they're not that dumb. And so for my work in this field, I wanted to get at some of these specific structures and figure out if you could predict the size, the relative size of a brain region, which you can see on this histological section from my co-author, Dr. Andrew Iwaniuk's lab, you could correlate the size of that structure in the brain with the size of the external structure on the brain endocast. And so one of the structures I looked at was the optic lobe overlying the optic tectum, which I pointed out way earlier when we were talking about this idea of relative size of brain structures correlating with behavior. The optic tectum receives visual information from the eyes, and it has been shown to be relatively smaller in things that might not be relying on their sense of vision as strongly as other birds. So things like owls, parrots, ducks, and geese. And so ducks and geese actually have a very well-developed sense of touch in their bills, so they might not need as much visual information. And the cool New Zealand birds like kakapo, which is a nocturnal flightless parrot from New Zealand, and kiwi, again, my favorite, that flightless bird with a cute long bill from New Zealand. And so I wanted to know, you know, if you just had a fossil where you could measure the optic lobe, but obviously don't get this internal structure for the optic tectum, can you predict the size of the optic tectum and therefore make inferences about the behavior in extinct birds? And so to get at this, I tried to look at a broad evolutionary sample of modern day birds. This is all the endocast of extant or modern day bird species I looked at here and put into context of their evolutionary relationships represented by this round tree. And I measured the optic lobe surface areas, which are highlighted in orange on every single one of these endocasts. And just for context, there's a little skull down here in the corner that has the, well, I shouldn't point because I don't know if the camera slipped, but the beak is pointing to the left. That gives you orientation for the rest of the endocast. So all the endocast front is left on the slide. And you can see the optic lobes highlighted on the endocast of these 34 modern species. Then I ran a statistical analysis to look at the closeness of the relationship or the strength of the relationship between the volume of the brain structure, so the optic tectum, and the surface area of the endocast structure, the optic lobe. And so each one of these points, these round circles, represents a different specimen in my sample. And you can see that they're all clustering really closely to this trend line. And so there's a really close and strong relationship between this brain, the size of the brain structure, and the size of the endocast structure. So that means that at least in this case, we can use the size of the endocast structure, which we can get in fossils, to predict the relative size of the brain structure, which then has functional or behavioral implications. And so I took that information and plugged in some fossils into my data set. So on this plot, all of the open circles, so all the white circles with a dark line around them, are the modern day birds, and all of these filled blue circles are the fossils. And this is showing the relationship of just that brain structure, so the optic tectum volume, to the overall brain volume. So again, talking about the relative size. You can see that most of the fossils kind of fit within the pattern that we see in the modern day birds. You have a few up here. That's because a lot of extinct birds ended up being much bigger than modern day birds. So being up here just means that they were generally bigger bodied. But you can see there's like this one weird extant outlier, this modern day species, and that's my favorite, the kiwi. So he's over there, and you'll also notice there's another weird fossil outlier. This is a species called Diornis robustus. It's the common name is the South Island giant moa. It's another New Zealand species. And you can see there's a little reconstruction of the skeleton next to a man. So they were really big, also flightless. And so like the kiwi, they're not as extreme in their reduction of the optic tectum, but they are significantly different from the rest of the sample. And so, like I mentioned earlier, the optic tectum is relatively smaller in birds with reduced visual systems, like the kiwi. So to give you a specific example about kiwi, like I said, they have really touch-sensitive bills, a really good sense of smell that they use to find their prey. So they don't need a lot of information from their eyes. And in fact, there's a population of kiwi in the wild that has started to go blind. So they've started to lose their vision because they are so not reliant on it. And if you look at the endocast, you can barely even see the optic lobe on there. There's like that little sliver of orange under there compared to the size of the optic lobe of the rest of the birds on this sample. So you can visually see in the anatomy the same trend that we were seeing when we measured the anatomy. And like I said, we also had that weirdo Diornis of the moa. Now you can see the optic lobe isn't quite as small as the kiwi, but again, it's relative to the rest of the brain, pretty small compared to the rest of the birds in this sample. And again, that was borne out by our data. So to me, you know, numbers are great. We need statistical analyses and quantitative measurements, but it's really cool to be able to see it in the anatomy too, because the anatomy is the part that I like a lot. I feel like being able to see the difference makes it a lot more impactful. And so with this method, you know, the method of confirming that there is a relationship between the size of the brain structure and the endocast structure, and then using that to predict the size of the brain structure in extinct birds, I can go from the fossil of an extinct bird. So here's a reconstruction of a Diornis skeleton from the Carnegie Museum of Natural History. I can make a model of the soft tissue anatomy that is no longer preserved in the fossil record, like this endocast here, and use that to kind of recreate a picture of how these animals lived. And so in the case of the moa, those reduced optic lobes could mean that they didn't really have a great sense of vision. And some of this research is highlighted right now in an exhibit at the Science Museum of Minnesota called Science Superheroes. So in case anyone's tuning in from the Twin Cities or Minnesota, if you want to learn more about this, you can go check that exhibit out. And so when I do science, I like to share it with people, obviously through methods like this. Another thing that academic researchers do is share their findings in academic journals so that other scientists could then take my method and apply it to different extinct birds. So both of these have been published in these two different articles. And with that, I'm going to entertain some questions. First, I wanted to acknowledge my co-authors, so the people who helped me do that research and write it up in those articles, Dr. Lawrence Whitmer, who was also my PhD advisor, Dr. Andrew Iwaniuk, and Ryan Ridgely. I would like to thank a couple people who helped me out with the analyses, like Dr. Haley O'Brien, Dr. Daniel Sepka, and Dr. Ryan Felice. Special thanks to some of the undergraduate workers who helped me make those endocasts, so Meg Wakui and Shelby Jordan. And also, none of this science is possible without funding. So I was lucky to receive funding from the National Science Foundation Graduate Research Fellowship, as well as grants from the American Museum of Natural History Frank Chapman Award, so that's for bird research, and through at my PhD institution, Ohio University, there were grants through the Graduate Student Senate, and through the Office of the Vice President for Research and Creative Activity. So I really appreciate the funding from those sources. Um, and also, also none of this would be possible without the museums that loaned me specimens from their collection. So for this research, I used specimens from the American Museum of Natural History in New York, the Field Museum of Natural History in Chicago. I visited the Yale Peabody Museum in New Haven, Connecticut, to look at some of their fossils, and also things from the Ohio University vertebrate collections where I was doing my PhD, and the Smithsonian Institution National Museum of Natural History. So with that, I'm ready for questions. Gabe, you can stop sharing my screen if you want so you and I can chat and I can take audience questions.
All right. That looks good. Let's see. All right. Well, thanks, Catherine. That was really, really cool. Sorry. There. Was was this something that you've always wanted to study as you were going through your career?
Um, I knew I wanted to work with animals. So growing up, I thought I wanted to be a vet because I thought that was like one of the careers you could have if you wanted to work with animals. I knew about zookeepers, but I don't know. Somewhere along the line, I guess I internalized the idea that maybe vets make more, or there are more vets than zookeepers, so it's easier to get a job. So I thought I was going to be a vet. And then I went to undergrad, and within my first year of coursework, where I took a few pre-vet courses, I realized like, it's not really what I want to do. But didn't really know what I was going to do next until I took a dinosaur class my second year. It just sounded interesting, so I figured why not. And that's where my undergrad research mentor, he was teaching it. So I took that one, really liked it, asked him if he was teaching anything else, and he was. So I took the next class and then the next one. And then he actually had a grant to pay a student to do research in his lab. So he invited me into his lab. And I think that's what really sent me down this track. So my interest in animals led me there indirectly, but no, I definitely didn't like grow up thinking I was going to be a paleontologist or evolutionary biologist or ornithologist.
It only takes like one spark usually, right? Like just that one thing to get you going down the rabbit hole or, dinosaur
Mm-hmm.
hole, I guess.
Yeah.
I think one of my favorite things about your kind of research is when people think about paleontology, we only think like, just the bones, right? It's like what you can you see and what can you get. But for yours, you're kind of literally filling in a blank spot in in the fossil and like learning about the soft squishy things that we normally can't and that people don't usually think of when we're talking about paleontology.
Mm-hmm.
Um, do you think the reason that you're able to do a lot of this research is it like the advancement in technology, or is this actually a field of research that's been going for a while?
So it has been going for a little while. There was recently a paper that did kind of a historical review of the field. Um, but interestingly, one of the early papers about this turned out to not be true. Like what someone who first described one of the endocasts of, I think it was Ichthyornis, a later paleoneurology worker found out that he like hadn't actually based it on any real fossil material. It was just like his ideas of what it would be.
Ooh.
So it has been going on for a while, but maybe not super honestly at the beginning. But the researcher who showed that that first paper maybe wasn't super accurate, Tilly Edinger, was an amazing German scientist, woman who actually had to escape Germany before World War II because she was Jewish, and so she came over to the U.S. and she did a lot of really important work in the field. She did some birds, a lot of mammals, and really advanced paleoneurology. And there are a lot of other people who were working on it, in between and after that. But her story is one that's always really stuck with me ever since my friend in graduate school, Dr. Ashley Morehart, introduced me to it. But should say that all of the people who were doing this before CT scanning happened had to rely on endocasts that occasionally you get really lucky and the endocranial space gets filled in by sediment that is harder than the bone, and so then the bone of the brain case erodes away and you're just left with a natural endocast. So before CT scanning, people could use those, or if you had a fossil that had an empty brain case, you could paint in lots of layers of latex, so like a soft rubber, and then peel out the endocast through the back of the skull, basically, the hole where the spinal cord passes. So there were ways to do it with physical endocast, or if you were willing to cut it in half, you could see it. But we don't always like to do that to our fossils. CT scanning is non-invasive, obviously, because we do it to living humans. And so you don't really need to do much to the fossil. I mean, if it's in a giant block of matrix, you might need to prep it down so you're just getting the region of interest so it'll fit in the scanner. But it's a method that's really opened up these kinds of studies in lots of different specimens, whereas before we were limited to ones that happen to be preserved right or we were willing to sacrifice.
Those are the really cool kind of like advances in technology that I just love to hear about. Because
Yeah.
I can't imagine like taking the time to like paint latex inside of like a tiny like I don't know like Archaeopteryx or something, or even a Velociraptor, and just pulling that out and hoping it doesn't tear.
Yes, yeah. And if you don't leave it behind inside there, oh, yeah.
And then like the potential damage as a collections person, just the idea of like
I
I
know.
can't. It's too scary. Okay, well, let's look at some questions that we have from our audience. Let's see. One of the first questions we have is from Jared McGowan, asking if modern avian brains have about twice the density of neurons as a mammal brain, what can we infer about the internal structure neuron density of dinosaur brains from them?
Ooh, so this is a great question, and demonstrates like a pretty up-to-date knowledge of the field. So we can't just make inferences about non-avian dinosaurs from birds. For avian dinosaurs, like for extinct birds, yes, we can make inferences from their modern day relatives. But birds are not the only modern day relatives of dinosaurs we have to look at. We also have crocodilians. And so dinosaurs, like evolutionary speaking on the tree, they're kind of wedged between birds and crocodilians. So like you have crocodilians over here, birds over here, and then here's the dinosaurs. And so if we want to make strong inferences about what dinosaurs did, we have to look at both sides of their extant relatives. Um, and so I don't know if anyone has actually looked at the packing of neurons or the neuronal density in crocodilians the way they have in birds and mammals. So to get at this question, that would be the first step. And then, you just have to figure out how strong your imprints can be and how much you would lean towards crocodilians or birds. There's a lot of much more advanced computational and statistical methods that can let you kind of weight those decisions based on the relatedness. But yeah, we need the information from the crocodilian side before we can really say if dinosaurs were more like these super thinkers like birds with the highly packed neurons, or they were maybe doing more things like crocodilians, which I'm not going to say they're not super thinkers because we don't know yet, but no one's demonstrated it the way that our understanding of bird brains has grown a lot in recent years in ways that our understanding of crocodilian brains has not.
We don't want to offend any crocodilian lovers out there anyway.
No, no. And I lived in Florida for a year, and those alligators are so cool. Like, I don't want to diss them, but we just don't know.
Uh, when I visited University of Florida a couple times, I wanted to see alligators so bad, and I didn't see one. I only saw one on the very last day that I was there, on the last time I went, and we had to go like to one of the reserves just to see
Hmm.
it, and I was like, I thought they're everywhere here.
They or like when I would need a stretch my legs break, I would just walk out of the office, and there were like some little woods areas, actually probably a like drainage ditch, and I could almost consistently see an alligator in one of those. So you just got unlucky.
Oh man, I guess crocodilians don't like me.
Maybe, maybe. Well, they know you're a collections person, so they don't want to be added to the collection.
That's true. That's probably true. A really on another side note for that, like there's the old expression like bird brain, right? Meaning not quite very smart. But from your research and what you're talking about, it seems like birds aren't actually bird brain. They're very high thinkers and things like that. What is it like? What is the evidence in there that besides like actually seeing them, like that can really point to that? And why is that misconception of bird brain?
Well, I'll start with the misconception. I think we do this a lot, which is why I don't want to jump to say that crocodilians aren't as smart. We tend to define intelligence as like relative to what we have to do to get through life, like the tasks that our brains have to serve, and what we think intelligence is. But birds don't need to do the same things we do. Like birds don't need to go sit in an office and use a computer 9 to 5. They have very different demands on their brains and on their bodies, and so they have the right kind of intelligence for their function, just like other animals do too. Certainly, some of them maybe aren't as great as their functions and as others, but I think defining a human definition of intelligence is um, unfair and kind of limits our understanding of the different ways that brains work in the world around us. And so recently, there's been a lot of research looking at the internal anatomy of bird brains, so like from extant ones, when we actually have the squishy ones and we can cut into them. People have found that bird brains are just organized differently from ours. So ours are organized in layers, and theirs are more organized in a nuclear fashion. So like what might be a layer for us is like these chunks in their brain that all connect together. So it's just a different organization, but it's still a very good way of storing and processing information. And so I think that's where a lot of the misconception came from. It's because of the layered way that mammalian brains work, we have all these wrinkles on the outside of our brain, and birds don't have those wrinkles. And so humans just looked at that and assumed like, oh, no wrinkles means they're not very smart. Actually, they just evolved a different way of packing those neurons in and storing the information. So that's the newer research that's kind of dispelled the bird brain idea.
Awesome. And like if you're a kiwi, you don't need those parts for your eyesight apparently very well,
Yeah,
even though they've got tiny heads.
yeah.
I will say that video you have of the kiwi was just adorable. I love kiwi so much. I went to New Zealand, I think a few years ago, and we went to a kiwi like what's it called? Like a not a reserve, but like a kiwi reserve. And then
Hmm.
you got to go into like their dark room and you just watch the kiwis run back and forth, and I was just like, these are the cutest birds in the world.
They are.
They're
They're
also
so
much bigger than
special.
you think they are.
Yeah, yeah, and they can like, so my special kiwi story is that I've met a kiwi at the National Zoo in DC. They have one kiwi named Pip who is actually pretty into meeting people. Not all kiwis like people, and so you can like meet and pet Pip. Uh, and he's a little feisty. Like, I mean, he was fine with being petted, but like he was doing some jumping that was getting him into trouble. His keeper was like, no, no, we're not doing that. And apparently he could, and other kiwis can really pack a kick with those strong legs. So they look cute, but if they don't want to be around you, just like any wild animal, they can do some damage.
I am adding that to my list of things to do when it's okay
Hmm.
to travel again, because I would
Yeah.
love to meet a kiwi in person.
Yes.
Okay, so here's another question from the chat. If we are able to determine what part of the avian brain is devoted to processing audio information, are we able to identify the same regions on most or if any dinosaur endocasts?
Ooh, okay. So part of the problem is that those regions are kind of deeply buried in the brain. So I picked the optic lobe for my research. I also looked at another structure called the Volst that overlies the hyperpaleum. Point is, I picked those two structures because they show up consistently on the endocast. But like I said with that earlier question about intelligence, birds don't have all these folds on their brains that we do that can kind of like at least on human brains, we see like, oh, this between this fold and this fold is named this region, and we know what it's devoted to. We don't have those landmarks on the bird brain and bird brain endocast. Um, and that major auditory nucleus in birds is actually kind of like in humans, you've got on your brain, you've got like the visual spot and then the hearing spot. This is gross oversimplification, but you know what I mean. Like there's, at least for the receiving the information, you've got these two spots. In birds, they kind of tucked the hearing one up under the visual one. So the hearing nucleus is actually tucked up under the optic tectum. So you really don't get to see it on the endocast. So that's a long answer to no, probably not, because they just don't show up on the endocast super well. And the other problem with non-avian dinosaur endocasts is that they don't fill their brain cases a lot with their brain. So like I was saying with like fishes, they have a lot more space between their internal surface of their brain case and the brain. Dinosaurs are kind of like that too, just like crocodilians. There are some researchers who have worked to kind of figure out ways around that, but it's not going to be as simple of a correlation as my research could be, where you've got, okay, endocast, surface area, brain structure, volume, and find a strong correlation.
So this brought up something, a memory for me. Like, so I watched Pacific Rim like a few weeks ago, right? And
Okay.
in Pacific Rim, there's this line from an old, like, I guess, myth about dinosaurs, and that dinosaurs have a hindbrain, right? That they had like because they're so large, they needed a kind of secondary brain to control the size of their bodies, right? It's one of those old like science myths that people would tell. I remember learning it in school myself. But actually, now that I think about it, and we're talking about dinosaur brains and stuff like that, I don't know where that came from. Do you know where that came from?
I don't. So this is like they have a brain in their butts. That idea,
Yeah,
right?
yeah.
Yeah. I don't really know where that came from. I will say that as far as I know, all vertebrates have like a slight sacral enlargement in the spinal cord. So like in the sacral area, like between your hips, there's a slight enlargement. I could be wrong about the all, but like the sacral enlargement is a thing that we can see in some things. So like maybe that's where it came from, but that is not the same as a brain. Like it's still spinal cord and like doing the things that the rest of the spinal cord is doing, which is like carrying signals down to the rest of the body and carrying them back up to the brain where they get processed. So I don't know where the butt brain idea came from though.
It just brought up that memory. Remember we were
Yeah.
watching that thing and I was like, oh yeah, they used to say that. Is that real? And now that we're talking about dinosaur brains, I had the moment to ask it.
I mean, it's a fun idea, like, but it's almost like you're seeing dinosaurs as like, um, two people in those giant puppets, where like the front is one person and the back is another person who's just bent over. That's not dinosaurs. They were still an organism that evolved like many other vertebrate animals, and like have that similar plan of the connections between the thinking part and the rest of the body.
And none of them are as big as a kaiju anyway, right?
I don't know actually. I haven't seen scales of like Godzilla versus dinosaur, but
Hmm.
yes. Well, I guess I'm thinking of like a scale of Godzilla versus a city and dinosaur versus city. Yeah, I think you're right.
Yeah, that's probably right. I would probably like to see that scale myself one day, though.
Hmm.
That might be something I need to do. Here is another. Oh, I just lost it. Where did it go? This one's a little bit off. If you don't know the answer, it's totally fine. But I will. Someone asked the question, so I'll ask it. So King Cretaceous is asking, is it true that there are currently two species of Spinosaurus?
Oh gosh, I don't know.
Okay, that's what I thought. Sorry, King Cretaceous, we tried. Uh,
It's one of those I don't know, and I know enough to not wade into it. So.
that is absolutely fair. I mean, that's the thing with paleontology, folks. We're all very specialized in what we study. Um, there's a lot in the world of paleontology. For what was it, 3 billion years worth of the history of life. There's no way one person can know all of those things. And even in the world of dinosaurs, right? Like, there's millions and millions of years, tons of species and genera to like understand and different interactions. There's no way that a single paleontologist is going to know all of this.
No. One of my friends who also studied science for undergrad, we were talking about how like when we're asked to do outreach, or just like in general, like your non-science friend is like, I have a science question. I'm going to ask the person who studied science. And she's like, but they seem to think that we're like encyclopedias. Really, we're just like a very small notepad of information. And
Oh,
then
I never thought about it that
seemed
way.
very accurate. It's like it's a very small, very detailed notepad about a specific uh, area of research. And so I feel like it's really the science communicators who have that like broader base of knowledge and can answer not any question about science, but like lots more broad questions about science. Whereas, yeah, us researchers, we have to really, really dig down into the details to get anywhere with our questions. So we don't always have time to keep up with like the latest science controversy.
That's very, very true. I mean, for me, like I kind of do both sometimes, but even as
Yeah.
a science communicator, like there's only so much my brain can process along with doing like outside work things that like
Mhm.
if I dedicate all of my storage capability to paleontology, I'm gonna forget to eat sometimes, I swear.
Yes. And that's important.
So when like kind of on that, when it comes to like your research questions, like you said, what was it specifically about like endocasts and bird brains and dinosaur brain evolution that really got you to go down this research path? Like what was the big question that made you be like, I want to look inside a dinosaur or a bird dinosaur skull?
Well, I didn't have like a specific question other than, just maybe I didn't know it at the time, but I've since realized that, like, yes, I really like these structure function questions. Another way of, another term for that field is functional morphology. Um, and then, like I said, I got lucky and ended up in an undergrad research mentor's lab, Dr. Daniel Sepka, who already was kind of studying some of these endocast questions. And so when you're an undergrad, you don't always like have a clear idea of what research you want to do, and that's totally normal, or at least it was when I was growing up. Actually, I think maybe even though that was not that long ago, I feel like now a lot of undergrads like feel like they already have to have their own research ideas and get some publications out before grad school, which is a lot. But at least for me, it wasn't super expected that undergrads like know enough to develop their own research question. Um, and so Dr. Sepka had a few different ones in his lab that he had data for me to work on, and he let me pick like, okay, well, which one seems more interesting? And I was just drawn to a question at the time about bird brain endocasts and if we could see a signal for diving behaviors in bird brain endocasts and like the different types of diving. So you have like birds that dive underwater and then get around by flying, basically, like penguins, or things that are maybe more foot propelled divers, birds that are diving to different depths and from different heights. And so we had a sample of birds that are engaging in all these different diving behaviors, and we're looking for a signal in the shape. And we didn't end up actually finding a really strong one, but that led me to like, well, what else can we learn about bird brain endocasts? And so um, that was like you mentioned earlier, the spark moment. And then from there, I got really into these questions like, well, how do we know what we know, and how much do we know, and how well constrained are those inferences? And that's where we ended up with these questions about like, well, can you tell the size of an optic tectum from the optic lobe? Which, maybe doesn't sound as exciting as like a new species of dinosaur, but I think is important to kind of ground the field and make us know the limits of our knowledge.
Oh, definitely. I mean, you know, describing a new species is cool and all, but it's like I like to joke like and say like every fossil's worth a thousand stories, right? And
Mhm.
so every story that we are able to get out of these fossils and tell fills in the gaps in those chapters of the history of life on Earth, right? Like as most of us paleontologists know, the fossil record is incomplete and we'll never be able to understand or see everything. But the more that we can fill in the gaps, the more we can better understand how interconnected everything is and how like how much how evolution like changed and experimented and everything to lead to the way things are and even help us understand kind of future things too, right?
Yeah, definitely.
So for the folks who had those questions that we may not be able to answer today, there's your question to kind of go down your own rabbit hole and do your own kind of research and maybe find the question for yourself. I mean, that's paleontology anyway for all of us at
Hmm.
home. I mean, I know there's a lot of times where like I see a new thing. I mean, later after this, I'm definitely gonna go into a Wikipedia um, expedition and start looking at more about brains for birds and stuff like that. So
And also looking up the scale of Godzilla to the biggest dinosaurs.
yeah, yeah, I'm
Yeah,
gonna definitely make a infographic about that one, and I'll post that later today.
right.
All right, well, I think that does it for today. Thank you so very much, Dr. Early, for joining us today. We really appreciate it.
Yeah, thank you for having me. This is a lot of fun.
So for folks at home who want to like learn more about your research or you or your museum, where are the best places for them to go to?
So if you search the Science Museum of Minnesota, you will find our webpage. We're still kind of in the process of updating. So I should say, I started there in the pandemic. I started like in, what is it, July, June, sometime during the summer. So we're still working on like getting me up onto the webpage. Um, but I still encourage you to check out the webpage because they have a lot of really great information about their other exhibits. They just actually redid their exhibit about race, and how it's just a historical construct and not actually a biological reality, and how that then influences people's lived experiences. So that's just been relaunched at the museum. They did the first one, which traveled a lot, and now they've updated it and will be traveling again. So they have information on their website about that exhibit and a lot of other equity-focused exhibits, which I like a lot. I think a lot of science museums don't make time and space for those. You can also, if you Google my name, you should find, I have a Wix site. So I don't know that it'll be like the top hit because I don't pay for like that search engine optimization, but you should be able to find that. So those are probably the best ways to check out my work. And I'm also on Twitter, actually, so you can follow me at Catherine M. Early.
Cool. I'll be sure to put those in the description. And the last question I want to start ending with for all of our guest paleontologists is if you could give like a quick piece of advice for folks who want to go down this path, maybe specifically about paleoneurology, what would you tell them?
Okay, I actually have two pieces of advice. So, first, if you decide that you're going to go down this path, I think the most important thing, like more important even than what research project you do for your dissertation, is finding good mentors. So, if you are looking at grad school, or even like undergrad, thinking about working with someone in undergrad, look into what they do, try to get to know them as a person, and see if it's going to be a good fit for you. Because if it's not a good fit, that's going to be worse than if you had a project that is like, yeah, kind of meh, because that's a long time investment. The second piece of advice is to really do your research on what your career goals are and like what you want to get out of life, because you can be really interested in paleontology and know a lot about it and never pursue a career in it. And that is perfectly fine. And I say that because as much as I love my job and I've been really lucky to have a pretty smooth path to get where I am, not always the smoothest, but like I'm where I want to be. I recognize that the job market for things like this is not always great. So please pursue your dream, but just before you invest a lot of time and money into degrees, make sure that there is the kind of job and the financial stability that you want at the other end of it. So practical advice, but I think important because there's always encouragement. Like, dinosaurs are so cool. No one's going to tell you like, oh, don't study that. But you also need the realism that you can make it, but it's going to be hard. And just make sure that that the way you decide to pursue paleontology is going to be a way that also lets you meet your other personal goals.
A very important advice. Thank you so much, Dr. Early. And thank you so much to all of our online audience for joining us today for our first featured speaker on the mighty dinosaurs for Discovery Day Online. If you like this program and want to support programs like it at the Alf Museum, you can find links in the description below on how to do that. And as always, make sure you like and subscribe to our channel for more stories from the world of paleontology. We will see all of you again at 1 p.m. Pacific time for our next speaker, Dr. Nate Smith, as we talk about Antarctic dinosaurs. Thank you all so much, and we'll see you later.